Hi, my name is what's my name?
Jeremiah.
Jeremiah.
And I'm Carter.
We're gonna be talking about my mitosis,
Meiosis.
meiosis,
Meiosis.
miso soup.
Okay, moving on. Meiosis is a type of nucleus division for sperm cells
Boy reproductivity,
and egg cells.
girl reproductivity.
I really like milk.
But
and
before we
cookies.
tell you about the
Does
details of
milk taste good?
meiosis, we have to
Also
get
like
the
when
other
a little
person in the
birdie
room to
brings
shut
it
up.
to me because it's not Santa Claus. And
How do cells prepare for meiosis?
I don't know.
In interphase, the time in between division is spent copying everything the new cell will need, like chromosomes. They go from this to that.
Duplicated.
Duplicated, yes. Very good. Like organelles, but most importantly, DNA. So the new cell will have a full set of genes. Now, meiosis can begin. It starts with prophase 1.
Hey, that's like the thing in like mitosis.
Shut up. So in prophase 1, the chromosomes become visible.
Take your time. We got all day.
Yeah, we do. Just like that. And it's obviously in the nucleus, which is starting to break down. And two centrioles, here and here, go to opposite poles and create spindle fibers. And will attach to the centromeres, the middle part of the chromosomes. And this is where the first layer of genetic diversity happens when they.
What's that?
What's what?
Genetic diversity.
It's what helps organisms in an environment be totally different from one another. Now, the first layer crossing over. It's when chromosomes swap some genes with each other.
Help me. This one. Boom.
Guess what happened?
So far, it seems that meiosis and mitosis are very similar.
Yeah, you would think so, huh?
What's that supposed to mean?
We haven't even scratched the surface. Let's continue to the description.
Yep, go.
During metaphase, the homologous chromosomes pair up and line up at the equator. Like that.
Yeah.
Yeah. Of the nucleus. What a homologous chromosome is, you may ask?
Yep, I'm asking.
They are identical chromosomes, same length, shape, and genes. At this point, the spindle fiber is attached to the center of the chromosome, or the centromeres. During anaphase 1, after metaphase, the spindle fibers pull apart the chromosomes to the opposite poles, like that. Two big things happen here. The homologous chromosomes separate, which doesn't actually happen in mitosis. The second layer of diversity happens here, random assortment, which is when chromosomes are randomly pulled to one pole.
So you're saying that this one could have been pulled to that side, and this one could have been pulled to that side, and it's just random.
I think so. In telophase 1, the cells start to diverge, and the membrane of the nucleus starts to reform. And finally, cytokinesis is when the cytoplasm divides, and there are officially 2 cells. So we're done then, Carter?
No, we're not, Carter. We still have to discuss meiosis 2.
Meiosis what?
Meiosis 2.
What 2?
Meiosis 2.
Meiosis what?
Meiosis 2. You know, the second part of meiosis.
Oh.
All right. Both of the 2 cells made at the end of meiosis 1 will now undergo meiosis 2. We're only going to focus on one, but just imagine that there's 2 happening with this. A note to make that these cells do not divide during meiosis 2, and that there's no DNA replication from meiosis 1 to meiosis 2. So, during prophase 1, chromosomes become visible again, and the spindle fibers form and attach to the center of the chromosomes.
Yeah.
And at the beginning of metaphase 2, the chromosomes line up at the center of the cell. And during anaphase 2, the sister chromatids get separated. And finally, in telophase 2, two new cells start to become apparent. And then after telophase 2, they both go through cytokinesis, resulting in two cells. And since we had another one going on, the other one divided, which would equal 4 cells in total at the end of meiosis 2.
Wow, this sure is complicated.
Yeah. So prophase 2 is similar to prophase of mitosis,
Yeah.
and metaphase 2 is similar to metaphase of mitosis,
Yeah.
and anaphase 2 is similar to anaphase
I
of
think they get the point. There are a few distinct differences between meiosis 1 and 2. The number in their names changes.
Aside from that, in anaphase 2, instead of the homologous chromosomes being separated, the sister chromatids get separated.
Another difference is that meiosis has the number of chromosomes in its daughter cells.
I think it's time for our math segment.
Oh no!
The original cell was diploid, meaning it had 2 sets of chromosomes. We'll use N as our set of chromosomes. But after mitosis 1, the chromosomes weren't duplicated again. So in meiosis 1, we had 2 new cells with just N. And then after meiosis 2, since they weren't duplicated in meiosis before meiosis 2, and 4 new cells with N over 2. To put that simpler, if we have one set duplicated to be 2, would give us 2. And then it was halved in meiosis 1, which would give us 1, and then halved again in meiosis 2, which would give us a total of 0.5, which is a half set. And since humans have 46 chromosomes, half would be 23 chromosomes total for each new cell at the end of meiosis 2. Any questions?
Are all the genes the same?
No.
Why is that?
Alleles.
Huh?
Alleles are differences in the same gene. So let's say you have gene 1, and someone also has gene 1, but they might have gene 1.1, which is the same gene, just slightly different, which adds another layer of uniqueness to people.
Like me?
Sure. So what's the point of having 2 different ways to divide nuclei? Wouldn't it be more efficient if all cells went through mitosis, and college biology students wouldn't have to find time to learn something else with their already crammed schedule?
I mean, that's kind of meta, but maybe. But meiosis allows for genetic diversity in population, and because the cells have half the number of chromosomes when they fuse reproduction, it ensures that the offspring will have the correct number of chromosomes.
Speaking of diversity, there are 3 layers of diversity, right?
Yep.
And we only have gone over 2.
Yep.
So what's the third one?
Random fertilization. When a sperm cell combines with an egg cell, both of those cells randomly fused when another from the pools of sperm and eggs could have fused. Well, I believe that wraps up our discussion.
Right, it does.
Goodbye. Carter, save some for me.
Are you proud of yourself?
Yes.